Carbohydrates are some of the most abundant biological molecules in nature. It's composed of carbon, oxygen, and hydrogen in the ratio CxH2OY. If we compare a glucose molecule to a molecule of water, we can see that the ratio of hydrogen to oxygen is 2 to 1 in both molecules. This is what the hydrates in carbohydrates refer to. Carbohydrates can be divided into monosaccharides, disaccharides, and polysaccharides, monomers of carbohydrates, as they are the simplest form of carbohydrates, containing only one sugar molecule. Because they're also classified as sugars, their names end in O's. They also are easily dissolvable in water. Monosaccharides can further be subdivided according to the amount of carbons present in the molecule. The smallest of these is the trioses that contain 3 carbons, for example, aldotriose. Others include pentoses that contains 5 carbons, like ribose and deoxyribose, and the hexoses that contain 6 carbons, like for example glucose and fructose. When these longer chain monosaccharides are dissolved into an aqueous solution, it will fold into a ring structure that is more stable. We can use glucose as an example. The carbonyl group of one end will react with a relatively distant hydroxyl group. The last carbon will not form part of the ring structure. Now this will result in 2 isomers of glucose, namely alpha glucose and beta glucose, can exist. The only difference between alpha and beta glucose is the orientation of the hydroxyl group on the anomeric carbon. In alpha glucose, it will point downward, and in beta glucose, it will point upward. Now, if you do not know which carbon the anomeric carbon is, it will be the carbon that acts as a stereogenic center. A stereogenic center is an atom that is related to 4 distinct atom groups. In glucose, it will be this carbon, as it's bonded to a carbon, an oxygen, hydrogen, and a hydroxyl group. Glucose has other isomeric forms as well, namely galactose, where the orientation of the hydroxyl group on carbon 4 is different, and also fructose, where the aldehyde is now a ketone. Monosaccharides are very important in biological systems as it provides a source of energy. This is because of the high amount of carbon-hydrogen bonds that, when broken, releases enough energy to produce ATP from ADP. Monosaccharides also serve as building blocks to provide larger molecules like RNA, DNA, and ATP from ribose sugars, as well as disaccharides and polysaccharides. Disaccharides are formed from 2 monosaccharides. The term di means 2. Now, the forming of a bond between 2 monosaccharides takes place as a condensation reaction. We will have a look at maltose as an example. Maltose is formed from 2 alpha glucose molecules. The 2 glucose molecules will be positioned by an enzyme, bringing the 2 hydroxyl groups alongside each other. Because of this, different disaccharides are possible. The 2 hydroxyl groups will combine, producing a glycosidic bond and then releasing water. The reverse reaction of breaking the glycosidic bond is also possible as a hydrolysis reaction, where water is added. The glycosidic bond formed as a covalent bond, which is one of nature's strongest bonds. In this example of maltose, the bond is classified as a 1,4 glycosidic bond because it connects the first carbon of the glucose to the 4th carbon of the other glucose. The 3 most common disaccharides are maltose, sucrose, and galactose. Maltose, as previously mentioned, is made from alpha glucose molecules and can be found in honey, beer, and bread. Sucrose is composed from glucose and fructose and is seen as the transport sugar in plants and can commonly be purchased in shops. Lactose is formed from glucose and galactose and is found in milk. Between monosaccharides and disaccharides, these sugars can be grouped as either a reducing or a non-reducing sugar. Reducing sugars can act as a reducing agent, which will donate electrons during a redox reaction. Take the overall reaction for cellular respiration as an example. The glucose molecule will be oxidized to form carbon dioxide, and oxygen will be reduced by the glucose to form water. A reducing sugar can be distinguished by its anomeric carbon, having a hydroxyl group as in glucose, fructose, and maltose. The non-reducing sugars have no hydroxyl group on the anomeric carbon, just like in sucrose. Lastly, we have polysaccharides. These are not classified as sugars and will thus not be soluble in water. They can be classified as polymers because they are made up of thousands of repeating subunits of monosaccharides that are linked to one another by a glycosidic bond through a condensation reaction. This also classifies them as macromolecules. Now, polymers are important as storage and structural molecules. If we were to store glucose in its basic form in our cells, it will lower the water potential of the cell as the glucose molecules are soluble, causing water to enter the cell. This will cause the cell to burst. Glucose is also very reactive and would interfere with the cellular reactions. Because of this, glucose would need to be converted to a compact, insoluble, and inert molecule. For animals, this is glycogen, and for plants, this is starch. Now, starch is composed of two molecules, namely amylose and amylopectin. Both of these molecules are made by linking alpha glucose molecules through a condensation reaction. Recall that alpha glucose is less stable than beta glucose. So using an alpha glucose is necessary as starch needs to be able to be broken down later on to use it as glucose molecules for respiration. Beta glucose is not as easily broken down. So by linking the glucose molecules by a 1,4 glycosidic bond, we will achieve a very long unbranched chain of amylose. This long polysaccharide chain will then curve and coil upon itself into a helical structure that is more compact. Amylopectin, on the other hand, is much shorter and contains 1,4 as well as 1,6 glycosidic bonds. This will cause the molecule to be branched. Amylose and amylopectin then combine to form starch grains that can easily be seen in chloroplast, potato tubers, and seeds using a light microscope. Starch is only found in plants. In animals, a similar structure to amylopectin is found called glycogen. Glycogen molecules clump together to form granules that can be seen in liver and muscle cells. The last polysaccharide that we will have a look at is cellulose. Cellulose is one of the most abundant organic molecules as it is found in the cell walls of all plants. Alpha glucose will not be suitable to use as a structural monomer as it is too easily broken down by enzymes. Thus, cellulose consists of beta glucose. Beta glucose has a slow rate of breakdown and will make cellulose mechanically strong. Because the hydroxyl group on the beta glucose differ from that of alpha glucose, it causes every second beta glucose molecule to rotate 180 degrees to be able to form 1,4 glycosidic bonds. This forms a long chain cellulose molecule. Now 60 to 70 of these molecules will bundle together to form structures called microfibrils. The hydrogen and oxygen molecules between the cellulose molecules will form hydrogen bonds. These bonds are weak but has an additive effect because of the enormous amount of these bonds. This makes the microfibril molecule very strong. The microfibrils can further form hydrogen bonds between each other to form fibers. These fibers will run in opposite directions across 20 to 40% of the cell wall. Other molecules will then cross-link with these fibers to form a matrix that is fully permeable and has a high tensile strength, almost equal to that of steel.